Nothing? Okay. So microbiology is the science to study small creations. We call them microorganisms. And micro mean we will not be able to see them by naked eye. So you need an equipment or machine to help you to make them more, like to zoom them, to be able to see them. So micro mean too small to be visualized by naked eye. Okay. The microbe world that surround us is very diverse. Okay, we have different species and different, type of them, but they could be beneficial. At the same time, they could be harmful. Do you know how microbe could be beneficial to us?
Like intestinal bacteria?
Yes, intestinal bacteria. And that will bring me to tell you that we actually live in a very diverse world of invader around us. He said that we have bacteria in the intestinal, we have bacteria in the skin, we have bacteria in the air, we have bacteria in the soil that we walk and, everywhere. With all this diversity, how can we actually manage to understand and to study those, different groups? We need a system to be able to study those groups. The main, division on those microbes is the bacteria and the virus, the fungi, and the parasite. So in order for us to, I want to focus, sorry, to be able to see the horizon. Will you be able to see? Okay. In order for us to be able to study their role in our life, we need to have a system of classification. And without that system of classification, we will be overwhelmed. And there is no way to find where should I put fungi in relevant to bacteria. Okay, where my focus will be is bacteria. Fungi and parasites will be discussed after lecture 13 with Dr. Mahabbat, and virus will be discussed with Dr. Garwin towards end of the course, okay? So we will focus mainly on bacteria, and that will be the focus of my sessions with you. All right, back to the slide. So with all this diversity, we need to have a system, for classification, and without that system, it will be impossible, to study all details of these, microbes. Okay, the credit of the system of classification was given to a Swedish botanist named Carlis Linnaeus. Carlis Linnaeus was trying to make or minimize the chaos of having all this diversity. So what he did, he developed and credit for two achievements. The first one is the hierarchical classification system. Okay? Hierarchical classification system. So what do we mean by hierarchical? Do you know the word, meaning of hierarchical?
Level?
You arrange, yes?
Order.
From put them in a classes? Okay. So how actually you arrange, a large group in a small group based on what?
Similar characteristic features.
Similar characteristic. Okay. What more?
Genetic sequence.
Genetic sequence. Very good. Okay. More? Severities. Okay, that's yes, that's part, but it will come, later. Okay, so this guy, he put the hierarchical system classification, but he also did another thing. He did something called binomial nomenclature. So binomial nomenclature is a two-name system of, naming the microorganism. Okay, based on what? Based on a science called taxonomy. Okay, so what's taxonomy? In order for us to understand the basic of the classification based on taxonomy. So what's taxonomy?
Identifying and naming them different organic groups.
To identify and name them in different groups, yes? Based on what?
The characteristics they have, similar characteristics they have.
Okay, so taxonomy is a science of defining or identifying, the microorganisms and organize them, okay, in group based on what? Based on shared characteristics and put them on hierarchical, system of classification. So you have a large group, you actually look on this group, or say, well, these three are similar, you take them, put them aside, and after that, you arrange them on hierarchical classification from, highest to the lowest, okay? This is one of the achievements of Carlos Linnaeus, okay? So the second, why do we want to arrange or organize, identify them, name them, and at the same time, organize them based on the similarities? Why we should worry about that as a scientist?
Make it easy.
It make it easy, definitely. What else?
Link it to the diseases. Link it to the diseases.
Link it to the disease. What else? The most important, when we would like to have something, give it a name. Why do we, at birth, why do, family give like different names to the kids?
Be standardized around the world.
Exactly. So as a scientist, when we come to a conference, for example, and we talk about, Staphylococcus bacteria, if this is not the standard name, and I'm standing here discussing with different scientists from different parts of the world, they will not get what I'm talking about. So why? Because we want to have a consistency with the name, and that will help us to classify the organism. All right? If I go back, how the hierarchical classification look like? So the hierarchical classification starts with the domain, which is the domain is the highest one. Kingdom, phylum, class, order, family, genus, and species, and I have them in red because we'll have more discussion on that. So domain is the highest one, species is the lower one. So domain, we have, a gathering of group, but it's more broad of shared characteristic. The more you go lowest, you have more, close similarities between these, species, okay? So if I think of what Carlos Linnaeus done, he actually built this system from kingdom, okay? So domain come later. So he started from kingdom to the species, okay? So this is his hierarchical classification system. You start from domain, which is the highest one, the species is the lower one. The more you go lowest, the more, deep similarities between organisms. Are we clear? You understand? Confused? Overwhelmed? No? Great. All right, so the second part that Carlos Linnaeus is the nomenclature. And nomenclature means two name system. Okay, how he developed this two name system? The two name system is derived from the appearance of the microbes, the location, where they survive, and sometimes the name of the scientist who discovered that, microbes, okay? You remember I just highlight in the previous slide, genus and species. So this is the two-part name system, or we call nomenclature. All right, the genus is a group of organisms. They have, common traits, which is structure, biochemical, and physiological. So they share in these three, and they put them under what we call, genus. The species is the basic, taxonomic unit. So they share physiological and genetic characteristic. Okay? When we want to call a bacteria, for example, with the name of the bacteria, we use the genus and species. And there is a rule. So when you write the name of the genus, you start with a capital letter. You notice I highlighted on a different color. So the genus name starts with a capital letter. The species name starts with a lower, a small letter or lowercase letter, okay? Both should be italicized. So you don't have them straight. They have to be, italicized. But if you write them and you cannot italize them, you have to have underline on the genus and species. And this is you will face in the exam, soft system. if I ask you what is the causative organism for that specific, case, and you want to try Staph aureus, if you don't do the italics, you're getting 0 for that. So be sure if the system will not help you to do that, you do what? You underline the genus and the species, okay? All right, and you can also, abbreviate. I can say like S aureus, but the rule will continue. You have to have a capital start of the genus and lowercase of the species. All right? There is more system of classification, the 3 domain of life, and here they do classify microbe, and this is achieved to like Carl Wozius. The 3 domain of life was built on archaea, eukarya, and eukaryotic. So, yes, bacteria, archaea, and eukaryotic. Based on what? We talk about Carlos Linnaeus and his system of hierarchical classification and 2-name system. The 3 domain of life was built on similarity. So they did a comparative of sequence of ribosomal RNA, and based on this similarity, they divide the microbial world to 3 groups. Those 3 groups named as the 3 domain of life, or the 3 domain tree of life. Okay, it's bacteria and archaea and eukarya. So the 3 domain include these 3, but the 3 domain of life also, if you think of like domain, so what's after domain? Kingdom. Have 6 kingdom. So 3 domain and 6 kingdom. And this one we have until now. We still, deal with this classification. One thing I would like to highlight in these slides, bacteria and archaea, they have some similarities. This is why they gather them in one group and they call it eukaryotes. Okay? So if we want to go more, narrow in the classification, we need to understand the difference between eukaryotes and the eukaryotes. And eukaryotes have bacteria, archaea. In the kingdom, they have eubacteria and archaebacteria. And eukaryotes have the animal, plant, and fungi. And we have something called protista. And protista is anything other than animal, plant, and fungi. Okay? All the other group was gathered under that. And there is a lot of discussion about the protista. Well, they say there is some similarity, but then scientists say, well, there is a lot of this, difference among those groups gathered together. Okay? Recently, in 2015, they come a new classification for protista where they put protozoa under that group. Okay? Are we doing good? All right, so we reach eukaryotics and eukaryotics. So what's the difference between these 2 groups? The most important difference between eukaryotic and eukaryotic, if you look on the side image here, the cell structure. So the most important difference is the eukaryotics don't have what we call membrane-bound organelles. Do you know what's a membrane-bound organelles? Nucleus, mitochondria. Thank you so much. So this is the major difference between these 2 groups. But of course, there is a lot of difference. They are different in size. They are different in, the presence of nucleus and the absence. They different in the constituent of the cell wall. So eukaryotic have more, complicated or complex, chemical composition of the cell wall, while eukaryotic not. They have different in the cytoplasm. The eukaryotic don't have cytoskeleton, while the eukaryotic have. So this table will give you a summary of the major difference between eukaryotics and eukaryotics. Okay? Why we worry about that? Because when we go deeper, we would need to, based on this difference, we'll be able to do, different identification of those microbes. Why do we need to do identification of microbes? Why do we need to continue actually identifying microbes? Why? Yalla ya shabab. We're doing very well.
To understand the reason behind the diseases.
To understand the reasons of
Diseases.
Okay. So
we can treat them.
sorry.
We can treat them.
to be able to treat them. Okay, what more? You remember what Carl Linus did? He was trying to group the organism, you know. So when we actually encounter like, you know, new, any outbreak, we have like new, family member coming. And this family member need to be categorized and associated with a specific group. So this is why we should continue actually identify. We have to establish the microbe, key features. And after that, we have to associate them with group has previously, characterized, and get them connected to, to appropriate taxonomy, you remember, and assign them to their genus and species. And of course, doing all this, we will be able to study the effect of the pathogen when it comes to, like, if they have a beneficial or harmful effect on us. Okay? All right, moving to the next. So what next? How can we identify those pathogens? What are the methods that we could use to be able to identify pathogens? Do you have any idea on, in the microbiology, what are the theme or the method that we use to identify bacteria? No? Yes?
Whether you can see them under the microscope.
Yes. Can you remember when we introduce the microbial word to you? It's like we cannot see them by naked eye, so we need something to help us to be able to see them. Okay. What else? Stain. So stain actually will help us to visualize them under the microscope. What else? Shape. Okay. So when you say shape, can you remember we cannot see them by naked eye? How can I see? Shape under the microscope. Very good. What more?
Their genes.
Their genes? Nice. Okay, more.
In what environment do they survive?
In what environment they survive? Nice. And I think I don't need to finish the lecture. You guys are doing really well. Okay, more. Okay, so I can identify microbes with different tools and different methods. Okay, I did classification of these two different style. Don't also overwhelm with the information I get here, because the more we go, you will understand, getting back to the first lecture. So I can do what we call phenotypic classification or phenotypic identification. Phenotypic mean what? What mean the observation that you can see, okay? I'll tell you, in the lab, I got a sample from a patient and I wanted to know what kind of bacteria that cause the disease of this patient. I'll take the sample. The sample could be urine, could be blood. And I will culture. Have you heard the term culture? So you will culture. If you get an infection, go to the doctor, they will say, get your blood sample or urine to, lab. I'll culture it, and in a media, and the media is the environment where this, microbe can grow. I will do that, and I'll put it in a temperature, in incubator that suit the growth of suspected, pathogen, as with my experience. And I'll come the next day. The next day I'll have a small, tiny growth we call colony. So phenotypic classification or identification is to look on, this is one, is to look on how are those colonies? What are the size? Do they have a special color? And how they arrange the shape of the, the colony. I could also take the blade and smell it, because certain microbes have a very distinct smell that will help me straightforward to tell. For example, Pseudomonas, you will hear and you will study. Okay, so this is what we call macroscopic, examination. The other thing is what is microscopic examination, and microscopic examination, which you will discuss in the lab. I need something to help me. I cannot take the bacteria direct and put under the microscope and see. I need something to help me, so I'll use staining. Why? Because I can look on the general, but I need to look on details. So I'll take staining. The most famous one used is gram stain, and gram stain will help you to identify bacteria into two groups, gram-positive and gram-negative. Gram stain is connected to the cell wall of the bacteria. So whether bacteria is gram-negative or gram-positive is really rely on the constituent of the cell wall, which we'll discuss in the coming lecture, okay? So this is one thing that's, take me to another classification. I can also look or observe the ability of certain bacteria to kill RBCs in the media. We call it hemolytic, properties, okay? And that's also something you can look, if you look on the plate here, this one, it can tell you actually, this is like how the hemolytic results will appear. I don't want to go in details at this point. What we can do also, we can look on biochemical reaction. We can look on fermentation, and fermentation means ability of bacteria to ferment type of sugar like glucose, maltose, okay? I can look for the enzyme and protein enzyme produced by that bacteria, okay, or microbes. That's all could be part of the, phenotypic analysis. I can do what we call analytic analysis, that I can see the cell wall of the bacteria, the lipid, the fatty acid. This is more complicated kind of analysis, and it needs like special lab, it needs more manpower and stuff. The last thing, which, talking about is the genetic, or we call it phylogenetic. So phylogenetic analysis that include like, all the molecular techniques, like, PCR, look on the DNA extract the DNA from unknown organism, hybrid that with known organism, and see if you manage to, do identification. All right, all these methods, or especially the phenotypic, is like a starting point, which actually lead you to go more and do more tests to be able to reach, identifications of the bacteria. And of course, for the reason that we discussed, this is why we should continue, like, use different methods. And of course, like, we can just walk out today and someone will come with a new method and, more classifications, okay? All right, I'll give you an example. And the example here, gram-positive bacteria. How I know it's a gram-positive bacteria? I forget I have this, okay. So how I will know I have a gram-positive bacteria? Based on what I discussed with you, like stain. So the stain will take me to either gram-positive or gram-negative. And then under the microscope, like the shape, when I look on this, I can see either bacteria on a circle, that means we call it cocci, or a little elongate, we call it bacilli, okay? So that's something I could see under the microscope following the staining, and then the respirations, whether the bacteria is aerobic or anaerobic. So do they need to grow in the presence of oxygen or absence of oxygen, okay? I can go more and look on the shape or reproduction. So the bacteria under the microscope could be arranged as a single cell, okay, or cluster, or a chain. If they are in a single cell, you remember, so like it's cocci. If they are in a cluster, I'll call them staphylococci. Staphylococci. Just remember, we'll discuss that more. If they are arranged in a chain, we'll call them strep, okay? And of course, by this, we reach to identify the genus and species. And this is just an example. Again, we will have more discussion when we talk about, gram-positive cocci, or gram-positive and gram-negative. All right. Another example, if I want to look on the medical important, pathogen, here is like, another classification. So you can actually do your classification according to the physical properties of the cell wall. So you can have some bacteria that have a very rigid cell wall, flexible cell wall, or lacking cell wall. Are you aware of any pathogen that lack a cell wall? And those are the ones who make our life very difficult. Okay, so mycoplasma. Okay, you can also classify them based on the morphology. Some bacteria is just a simple unicellular. Some bacteria are, or microbes, sorry, is filamentous. So filamentous have, an organelles to help them to move. Some bacteria, some microbes, sorry, I'm just keeping saying bacteria, sorry. So microbes will live like, freely, but some are obligate intercellular parasite. So they cannot survive unless they are inside the host. Okay? And we come to staining properties, which is gram-negative, gram-positive, and after that, based on the arrangement and appearance under the microscope, we can just reach to the shape and of course, like oxygen tolerance, whether they can grow in presence and absence of oxygen. Okay? All right. We actually have all this discussion, and we come to another group. So we talk about fungi and parasites under the category of what? Eukaryotes. So what we discussed previously was eukaryotes, and now we're moving to eukaryotes. As I said, like I'm not gonna go in more details because this will be like I think like 6 or 7 lectures with details. But I want you to understand that the fungi under eukaryotes are actually grouped, can be live saprophytic or parasitic. What we mean by saprophytic? Yalla ya shabab, you're doing really well. I'm very proud of you. Yalla. What's saprophytic? New voices. No? Those are the microbe actually. Exactly. They live on that, organism. So they get their food from that. And parasitic, parasitic. So they actually live on another host and then they get their food. Okay. The fungi or fungi are divided based on the, sexual activity. So they can be or exist in a unicellular cells. And the unicellular cell example is yeast, and those are replicate asexually, okay, like by budding. Okay, the other form of the fungi is the filamentous. Example for that is a mold. You see when you have a bread in your kitchen and it's still like you're wrong, get old, and then you have this green stuff growing on it. So this is the fungi. Okay. They consist of thread, we call it mycelia, and that's elongate and branch. If you see the image here, this one. So this is like an example. And they can cause different type of disease. Again, I don't want to go in details, but just remember that they could cause superficial or subcutaneous or opportunistic, deep mycosis. So don't worry about this at this point. We'll discuss it more. Okay. If I think of the classifications of the fungi, which I just told you, they divided or classified based on their, sexual activity, like asexual or sexual. And of course, this is also, you're going to go in deep details when you discuss, mycology. But fungi is classified into 4 medical groups: common mold or Zygomycota, the sac fungi or Ascomycota, or club fungi called Basidiomycota, and the imperfect fungi is Deuteromycota. So imperfect, why imperfect? Because there is no sexual, activity was reported for that group. This we call imperfect fungi. Okay? All right. So what are we left with? We left with another group of eukaryote or protozoa. And protozoa, again, is classified into 4 groups. If you look on the table here in the top, you can actually zoom this table, so you can just make it larger. You will have a summary of the slides because it will help you to know if they have different names. I don't know why the screen, so it's just stuff from here. So these 4 groups is also, medically important. They call microsporidia or spore forming. We have the Fora, which and Apicomplexer, and Sacrocomostofora. So from the name, do you know how this classification are built on what? Their ability to move and ability to cure food. So this is why we have the spore forming. We have one group the ciliiflora move by cili. The AB complex is the one that know, organ of locomotion, and the Sarcomastigophora is the one that moves by pseudopodia or by flagella. So the classification will build on, their ability to move. Okay, beyond protozoa, we have the last group in the eukaryotic and called helminths. The helminths is, parasite, identified by looking under the microscope for an active form of them like larvae or eggs. So this is how we identify this group. And they are divided into two major groups, is nematodes, and that, roundworms, usually present in the intestinal of the human, and they transmit it through uncooked food. Okay? The other group is black helminths, which the flatworms include tapeworms, flukes, and also transmit through uncooked meat. Okay, if I want to look on this classification, this is how we put them. We have the black helminths and nemeth helminths, and the black helminths is divided into cystoda and trematoda, which will go in more discussion later. Okay, with the end of this classification, we wanted to know. We have the last group in the microbes is the virus, and virus is very unique. Why? Because virus is, they don't have this cell. So they are a genetic structure and coded with a protein, okay? Virus is obligate intercellular parasite. They cannot survive out. They need a cell to be able to, multiply inside the cell and cause a disease, okay? The virus have only DNA or RNA. It's not both of them. This is also unique for the virus. And surrounded by protein we call caspid coat, or sometimes they have envelope that's nuclear or cellular membrane drift, okay? Towards end in skin inside us, like in the gut, in the intestinal lumen, and women in the vagina, we have some bacteria, and where else? In the mouth, we have a lot of bacteria, trillions, billions of bacteria. Okay, so if I want to look on example of the amount of the bacteria we have, as I said, like we are surrounded in around and inside the body, we have in the skin almost near to 10 to the power 6 bacteria. In the mouth, we have, as I said, like trillion, like 10 to the power 11, and the intestinal lumen, okay, especially in the colon, we have almost 10 to the power 14 of, bacteria per gram, especially in the stool. Okay, so there is almost 0.2 kilos of the weight of the body is like, bacteria. Okay, all these bacteria and all the distribution, why they don't cause disease to us? I mean, they live inside us in all these different, with the amount they are huge. We're talking about millions and trillions. Why they don't cause disease to us? Yes?
They don't cause immune reaction.
They don't cause immune reaction, okay? They actually prime our immune system. This is why they live, with us. And based on that, I can tell you that those microbes who actually normally residing at a specific host size, that's us, okay, without causing any harmful effect, we call them normal microbiota, okay? So normal microbiota is all these bacteria live inside, host, and without causing any disease or any harmful effect. Okay? Those are opportunistic, because when they change their location, they actually could transfer and be an infective, so they can infect us. Okay? What I mean by that, the bacteria live in skin. If you just moving and have an accident, you have a wound or injured your hand, and this bacteria who normally reside in your skin get an access, that means they change what? They change their location. So at that stage, they can cause a disease, okay? So microbiota living at a specific site, reside there, they will not cause any disease, but then they could cause disease in certain conditions. For example, if they change their location, okay? There is certain organs in the body, they don't have any bacteria, and that you need to keep in mind, like your heart, your lung, your brain, your liver, your spleen, your kidney. Any pathogen change to that area will actually damage and cause disease, okay? All right, so what else other than normal microbiota? So microbiota, as I mentioned, could be beneficial, but it could be harmful. When it could be harmful other than changing the location? When? Do you think like you have all these bacteria, they live with you, we're friends, living good life together, but in certain conditions, other than changing location, they could be harmful. When?
When they decrease, if it's needed.
When they decrease.
For instance, bacteria in our skin, when it protects us, if the amount of bacteria decreases, that might have a side effect.
If the amount, logically, I can discuss that with you. Like, if you think the concentration of bacteria that live with us, if it decreases, okay, it can, get us like some kind of disease. This is what you said. Okay. Well, I cannot actually 100% agree on that, because the amount of the bacteria available, actually, they benefit from us without causing any harm effect. But if the bacteria, if you tell me like the concentration increase enormously, then the normal status, besides changing location, that could cause a disease. But there are certain cases like, they live with you and they prime your immune system. But what will happen if your immune system, get disturbed? This is when those, microbe can cause a disease. So if you have, certain condition, immunocompromised people, people who have problem with their immunity, they can actually get sick from those, microbes. Crossing into normal side, we already discussed that. And in case of infection, so when you get infections, that will also, be a source of the harmful effect of the microbiota. Okay, but at the same time, they have a beneficial effect, and I would like to discuss the beneficial effect on the last slides. Okay, so what's the process of the disease? To be able to have a disease. So what's the process? The process is to have a microbe, and this microbe, we call it a pathogen, okay? The pathogen is microbial agent. It's not enough. Must have pathogenic determinants. Those pathogenic determinants will help the pathogen or give them ability to enter, colonize, and multiply. Okay? So enter, and we will discuss throughout the course, enter, the certain factor will help the microbe to attach to specific site and to enter. So either reduced substance will facilitate the entrance. They need to colonize, and that's also, part of the, disease process. They need to multiply, be able to increase their number and concentration inside the host and within the susceptible host. Susceptible host could be someone having problem in their immunity or someone get infected, true? So this is susceptible. They need to find a suitable host will actually facilitate the process to happen. And of course, they have to overcome the immune response because the body will react. They need to have a possibility to overcome immune response. And when we discuss that, we talk about a pathogen that can, escape phagocytosis. This is how they actually overcome immune response because they will not be eaten, they will not be destroyed. So, okay, and they cause a disease. So if I ask you a pathogen, it doesn't just need to be a microbe. No, a microbe need to have pathogenic determinants. Pathogenic determinants like what? Virulence factor, okay? Those virulence factor, so the pathogenic determinant is the virulence factor will actually play a role, those are the, bad stuff in the bacteria will facilitate the disease to happen, okay? All right, so by what we discussed, I think we will be able to talk about what is the pathogenicity. So pathogenicity of the microbe is the ability of microorganisms to cause disease in another host or another organism, namely host. So this is a pathogenicity. So when I would like to define virulence, so virulence is the degree of pathogenicity. Are you connected? Pathogen need to have certain, specific feature, have, the pathogenic determinants, which the virulence. And if I would like to define what's pathogenicity, pathogenicity is the ability of microbe to cause disease in another organism or named host. And virus is the degree of pathogenicity caused by the organism possess virulence factor. Okay? And these organisms can actually invade the host and overcome the host immune response and causing disease. Okay. I would like to come to the last slide. So what's the importance of microbe in health and disease? There is, different benefit and harmful effects. But the benefit or the microbe could be beneficial to our, to us. Us could be like, human, but also they are beneficial to animal, to plant, to the environment. Okay, giving you an example, the human, we talk about normal microbiota and we talk about the effect. It could be like, production of some vitamins. They can actually prime our immune self. They can, occupy, area and compete for nutrition against other bad, organisms. Those all are, beneficial of microbiota to us as a human, but they can also be beneficial to animal by digestion of cellulose. Do you know what the digestion of cellulose? No. The cellulose. Sorry? Yeah, so it's a material in the GI of the animal, so certain bacteria will be able to digest the cellulose to help in the treatment of the animals. I said also it's helpful in industrial world because we can use bacteria to produce cheese, vinegar, wine, name it, bread also. So all these bacteria could be helpful. This is like in the industry. Environmental decompositions or recycling of organic compound, we call it sewage treatment, or bioremediations. So certain bacteria could be actually used to destroy or break down organic compound, and that could be helpful to the environment. Pathogenic, we talk, some microbe could be pathogenic to human, animal, and plant. And of course, when we talk about pathogen, we talk about clinical and medical microbiology. Those could also be, affecting our health, and part of the effect of this could help us also in go deep in the field of the infection and prevention control. Okay. وأقول قولي هذا واستغفر الله العظيم لي ولكم. This is the end of the lecture. I really hope that you guys get the general concept. If you have any concern, I would love to hear. from you now because we're almost done. We have 6 minutes. So I would like to hear what do you think about your first lecture? Is it broad? Did you get it? Oops, yes, of course.